In this video, I'll be doing some multiple choice questions, paper 1 from the IB. These are real past paper questions, so this is a perfect way to practice before you go into a test, so you know what kind of questions you're likely to get. So this is part 1. I'll make more parts to this. So I will be explaining these questions in some detail to be able to let you understand it better. I won't just give you the answer, so it will be more thorough. But before you do this, make sure to check out this video that I made, Transcription and Translation, that explains the process using a Bob the Builder analogy. So that will allow you to remember it far easier. So, where are amino acids joined together to make polypeptides? What I recommend is that you first write the answer you would think without looking at the options. That way you won't get confused when maybe the options aren't as what you thought. So give it a go by yourself first, then I'll give you the answer. So amino acids are joined together if you had to look at a cell. Amino acids are joined together outside of the nucleus. So this is the nucleus. This is where transcription happens. In translation, we know it happens outside of the nucleus, after the mRNA has been made. Now, the amino acids are joined together specifically on ribosomes. Ribosomes are these little dots, as you can see right here. They are either on this organelle here, the rough ER, or they are just floating free in the cytoplasm. This image doesn't show that, but these dots can also be floating around in the cytoplasm. So they're called ribosomes. So this is the site where amino acids are joined together to make polypeptides or proteins. Proteins your body will use to carry out a specific function. So the answer will be D. It is not nucleus, because nucleus is where transcription takes place, not translation. And we know in translation this process happens. Nucleolus is just within the nucleus. It's the dark zone within the nucleus, so this spot. So it's still in the nucleus, so this would not be correct. And Golgi apparatus is not correct, because after a protein is made, so after this process happens, so a polypeptide is just like a protein basically. So after a polypeptide or protein is made in the ribosome, it is then sent to the Golgi apparatus to be modified and packaged, kind of like to double check if it's okay before it goes to other places. So this is like the step that happens after the question. So next, on which molecule is a codon found? On which molecule is a codon found? So again, try it by yourself. So a codon, for those of you who don't know, this step is involved in translation as well. So in translation, we know the whole purpose is to make an mRNA, right? mRNA. So the mRNA is a sequence, basically a small part of DNA. And we know it contains a bunch of letters. It's like a book. It contains letters that are going to code for something. It's going to code for a specific protein, right? Because we're trying to make a protein. Now, in English, our words are different lengths. Some are long, some are short. But in mRNA, all the words are the same length. 3 letters. 1, 2, 3. So every 3 letters, every subsequent 3 letters is a different word. And in DNA, we call words codons. So codons is basically a sequence of 3 bases next to each other. So AUG would be a codon, AUC would be a codon, UCG would be a codon, UAA would be a codon. So remember, they're on the mRNA. They are not on tRNA, because tRNA will have the anticodons. tRNA will have the anticodons that will bind to the codons of the mRNA. Again, if you don't understand what I'm talking about, you have to check out the video for this to make sense. And rRNA, for those of you who don't know, this is like this stands for ribosomal RNA. So this is the RNA that will go and make a ribosome. tRNA is what will bring the amino acid, and mRNA is the code. A polypeptide is what we're trying to make. So a polypeptide does not have the codon on. Therefore, the answer is B. Let's move on to the next one. This is a very similar question. What is a codon? So this one was asking, where can a codon be found? This is, what is a codon? So give it a go by yourself. So option A is saying a sequence of nucleotides on rRNA that corresponds to an amino acid. So we know we can immediately eliminate a whole bunch of these just by knowing that a codon is not on rRNA, it is not on tRNA. So we can already eliminate A and C because we know it's on mRNA. But let's see D first. So likely it would be B. Likely. We'll first check D though. So it's a sequence of nucleotides. So if you don't know what nucleotides are these letters. These letters with their backbone. This orange line here is like the backbone. So it's a nucleotide is the letter plus the backbone. That's a nucleotide. So it's saying a sequence of nucleotides on mRNA that corresponds to an amino acid. So that's true because it's a sequence of 3 nucleotides corresponds to a codon. Now let's look at D. A sequence of nucleotides on DNA that corresponds to an amino acid. So that's not true. We know it's not on DNA, it's on RNA, specifically mRNA. And we know mRNA comes from DNA. So we get mRNA after transcription of DNA. Therefore it's B. Let's do one more. What is formed during transcription? So during transcription, try this by yourself first. So remember, during transcription, that's the first step. That's the step in which we're trying to, I have a nice diagram here. That's the step in which we're trying to turn the paper into the final product. So the rocket in my Bob the Builder situation. We're trying to turn the paper along, so the instructions, along with some tools and some materials, into the final product. So in the cell situation, we're trying to turn the mRNA along with ribosomes and tRNA into the final protein. Again, if you don't get this analogy, you need to watch this video. So the answer here would be, we need an, let's see, is it A? RNA strand complementary to DNA strand, formed by RNA polymerase. So this could be true because we know we're forming an RNA, specifically we're forming mRNA, but they're not specified here, so I don't think that matters. Just know that it's an RNA, and we know it's complementary to DNA strand because it's being copied from the DNA strand, so it's complementary. And it's formed by RNA polymerase. RNA polymerase is basically basically like the photocopier. It's gonna this enzyme is gonna photocopy a part of the DNA to give mRNA. So this is the enzyme that you're gonna need. Without this enzyme, this photocopying can't happen. So therefore I know it's this one. We know we're not making DNA, so this therefore immediately we can eliminate B and D. We're not making DNA, we're making RNA in transcription. Why is it not C? So because C is saying we're making an RNA strand complementary to RNA strand. No, that's not true. We're making an RNA strand from a DNA strand, not an RNA strand. And that's the first reason it's wrong. Second reason it's wrong is because they say using DNA polymerase. That's not true. We're using RNA polymerase. Okay, so that's why these are wrong and A is right. So I'll make a part 2 next, covering a bunch more of these multiple choice style questions. This is, in my opinion, the best way to study for a test after you understand the content, because then you understand how the IB works, and you understand what kind of questions they ask, because you have to understand that only a certain amount of questions can be asked. After a while, these questions will repeat. So if you practice enough multiple choice questions, nothing on the test can surprise you. So I really recommend you keep watching these kind of videos and keep practicing individual multiple choice questions.